Okay, let's dive deep here. So imagine, right? Your heart suddenly starts racing. Maybe your palms get clammy. That knot in your stomach. You didn't choose to make any of that happen, did you?
No, absolutely not.
Or, the flip side. You feel calm after a meal, everything
Yes.
just working away quietly inside.
Exactly. And that's the autonomic nervous system, the ANS. It's like the body's automatic control center for, well, most of those internal visceral functions.
Right.
And when we talk about changes, they can be incredibly fast. The source material we're digging into today really emphasizes the speed. Your heart rate, it can double in just, 3 to 5 seconds?
Wow. That fast?
Yeah.
Yeah.
Or blood pressure doubling in maybe 10, 15 seconds. Or going the other way, it can drop so fast you faint in just a few seconds.
Okay.
Things like sweating can kick in almost instantly.
Yeah.
Or involuntary bladder emptying that can be triggered really quickly too. That kind of rapid, powerful response is really a signature of the ANS.
It's like this hidden conductor, isn't it? Running things like blood pressure, digestion, how things move and secrete stuff, body temperature through sweating, bladder control.
Yeah.
So much stuff.
Constantly. It's running the show, adjusting our internal environment without us needing to, consciously think about it at all.
And the source material we have for this deep dive, it's described as this really detailed look, almost like a user manual for the ANS.
It really is. Very foundational. It lays out the structure, the wiring, the chemicals involved, the specific effects, with a lot of precision.
So our mission today, for you listening, is to really unpack this complex diagram and instruction set. We want to pull out the absolute core knowledge, stuff you really need to get a solid, robust understanding of how this automatic system works.
Yeah, the kind of understanding that, professionals in the field build on. We're going to go deep into the details that are in this source, explore the mechanics, and kind of see what it all means.
Think of us as your guides, helping you navigate this internal landscape.
Right. We'll point out the key concepts, try to explain the why behind things, connect the dots so you can really master this material, build that solid foundation, like the pros do, but hopefully in a way that's clear and, interesting.
Okay, let's get into it then. Let's open up this source material. What's the absolute starting point? What is the autonomic nervous system, according to this text?
Okay, so right at the beginning, the source defines it as the part of the nervous system that controls most, not necessarily all, but most of the body's visceral functions.
Visceral meaning internal organs.
Exactly. Things associated with our internal organs. So regulating your heartbeat, the constriction or dilation of blood vessels, the movement in your stomach and intestines, gland secretions, kidney function, bladder function. The list goes on. It might control some things entirely, but for others, it's providing really crucial adjustments.
And like we said at the start, that incredible speed and intensity is a key feature. Why? Why does it need to be so fast and powerful?
Well, it really boils down to survival and being able to adapt instantly. Think about those moments of sudden danger or needing intense physical effort, like right now.
Fight or flight.
That's the classic example, yeah. Your body has to reprioritize immediately. Blood needs to shoot to your muscles, away from your gut. Heart rate, blood pressure, they need to jump up to deliver oxygen and fuel. Metabolism has to ramp up.
Okay.
The ANS makes that massive shift happen instantaneously without you having to consciously process it. If you had to think, okay, constrict these blood vessels, dilate those, speed up heart rate, well, we wouldn't be very effective, would we?
No, we'd be toast.
Yeah.
So it's essential for emergencies, but also just fluctuating daily needs.
Absolutely. Responding to challenges, big or small. It's both an emergency system and, like you said, a kind of sophisticated internal climate control, all running on autopilot.
So where does this system actually get its orders from? It's not the conscious thinking part of the brain, is it?
No, generally not directly. Yeah. The main control centers are deeper, in parts of the brain and spinal cord that handle fundamental regulation. The source points to the spinal cord itself, the brainstem, which sits just above the spinal cord, and the hypothalamus, which is a really crucial control hub deep inside the brain.
These are areas managing basic survival stuff.
Right. Things like breathing, heart rate, body temperature, basic drives. That's where the primary ANS command centers are located.
But the source mentions some influence from higher up, too. Like the thinking brain isn't totally disconnected.
That's a really important point. While the core operations run from those lower centers, higher brain regions do have a significant say. Parts of the cerebral cortex, especially what's called the limbic cortex.
The emotional brain.
Yeah, exactly. The part involved in emotions, motivation, memory. It can send signals down to the spinal cord, brainstem, and hypothalamic centers and really tweak their activity.
Oh, okay. So that's how, like, feeling really scared or anxious can make your heart pound or your stomach churn.
Precisely. That's the neural connection. Your emotional state directly plugs into the ANS control centers via these pathways. It's a key part of that mind-body link.
Makes sense. And how does the system typically work on a moment-to-moment basis? Is it just waiting for commands from these centers?
A lot of its day-to-day operation relies on what the source calls visceral reflexes. This is really core to its automatic nature.
Reflexes like tapping your knee. But inside.
It's kinda, yeah. You've got sensory receptors scattered throughout your internal organs, constantly monitoring things like how stretched the organ wall is, blood pressure, chemical levels.
Like your stomach stretching after you eat or your bladder getting full.
Exactly. Those signals, which are usually subconscious, travel inwards to autonomic ganglia or up to the brainstem or hypothalamus. These centers process the information, again subconsciously, and then fire off an automatic reflex response back out to the organ to adjust its function.
So it's a constant feedback loop. Checking and adjusting, checking and adjusting.
Right. All happening automatically, regulating things without you even being aware of most of it.
It's like this internal self-regulating network. And the signals going out to the organs, the efferent signals, the system is split into two main branches, right?
Yes, the two big players highlighted in the source. The sympathetic nervous system and the parasympathetic nervous system.
The accelerator and the brake.
That's the common analogy, yeah. They often have opposing effects, but sometimes they work together, or one is just more dominant for a particular function. To really get them, though, you need to understand their wiring, their anatomy. Where do the nerves actually go?
Okay, let's trace those wires. Let's start with the sympathetic system's anatomy. Where do its nerves originate in the central nervous system?
Okay, sympathetic fibers start in a specific spot in the spinal cord's gray matter, the intermediolateral horn, and they only come from spinal cord segments T1, that's thoracic segment 1, down through L2, lumbar segment 2.
T1 to L2. That's it.
That's the origin zone. Because it's limited to these thoracic and upper lumbar segments, it's often called the thoracolumbar outflow.
Got it. Thoracolumbar. And from there, where do these fibers go? The source mentions they leave the cord and go into something called the sympathetic chain.
That's the first major step. So the nerve cell body is in that intermediolateral horn. It's fiber, called the preganglionic fiber because it's before the ganglion.
The first neuron in the chain.
Right. That fiber exits the spinal cord through the ventral root, joins the main spinal nerve for just a tiny distance, and then branches off. It travels through a little connecting nerve called a white ramus.
White ramus, okay.
And that leads it into one of the junctions of the sympathetic chain.
And the sympathetic chain itself. Those are the two chains of ganglia running down either side of the spine.
Exactly, like two strings of pearls running vertically next to the vertebrae. They're connected to the spinal nerves at various points. The source has a figure, 6-1-1, that shows this, and also shows other ganglia that are further out, called prevertebral ganglia.
Prevertebral, like in front of the vertebrae.
Yeah, things like the celiac ganglion, superior mesenteric, inferior mesenteric. They're located more out in the abdomen and pelvis, closer to the organs they supply. Nerves then go from those ganglia out to the final target organs.
Okay, back to the preganglionic fiber. It enters the sympathetic chain ganglion through that white ramus. What happens next? Does it just connect there? The source says it has options.
It does, and this is really key to how the sympathetic system can have such widespread effects. Once that preganglionic fiber is inside a chain ganglion, it can do one of three things, according to the source figure 6-1-2 shows this too.
Okay, option one.
Option one, it can synapse right there, connect with the second neuron, the postganglionic neuron, in the same ganglion it just entered.
Makes sense. Option two.
Option two, it can travel up or down the sympathetic chain itself, like climbing up or down that string of pearls, and then synapse with a postganglionic neuron in a ganglion at a different level.
So a fiber starting in the chest region could travel up the chain to affect something in the head?
Exactly. Or travel down to affect the legs. This allows signals from that limited T1-L2 origin to reach the entire body. It's crucial for distribution.
Okay. And option three?
Option three. The preganglionic fiber can pass straight through the chain ganglion without synapsing at all. It just keeps going outward, typically in a nerve branching off the chain, like a splanchnic nerve.
And then it synapses.
Yes. Then travels out further and synapses in one of those peripheral or prevertebral ganglia, like the celiac ganglion we mentioned, which are located closer to the target organs in the abdomen or pelvis.
So the second neuron, the postganglionic one that actually touches the target organ, its cell body could be in the sympathetic chain or way out in one of those peripheral ganglia.
Correct. And that two-neuron structure, preganglionic then postganglionic, is fundamental to the autonomic system. It's different from, say, the motor system where a single neuron goes all the way from the spinal cord to your bicep muscle.
Right. That synapse outside the central nervous system is the defining feature. Now, the source also mentioned something interesting. Sympathetic fibers ending up inside the regular nerves going to our muscles and skin. How does that happen?
Yeah, this is a bit less obvious, but really important. After the preganglionic fiber synapses in the sympathetic chain, many of the postganglionic fibers, the second neurons, don't just head off to internal organs. Instead, they loop back around.
Loop back.
Yeah, they leave the sympathetic chain ganglion and rejoin the main spinal nerves. They do this via different connecting branches called gray rami. And this happens at all levels of the spinal cord, not just T1L2 where the preganglionics originate. Figure 6.1-2 shows this loop back.
Wait, so preganglionic fiber leaves the spinal nerve via a white ramus, goes into the chain synapses. Then the postganglionic fiber leaves the chain via gray ramus and goes back into the spinal nerve.
You got it. White ramus in, gray ramus out, and back to the spinal nerve. And these postganglionic sympathetic fibers that are now traveling within the spinal nerves alongside sensory and motor fibers are small, unmyelinated fibers called type C.
And since spinal nerves go everywhere.
Exactly. Since spinal nerves distribute to basically your entire body surface, your limbs, etc., these sympathetic fibers hitch a ride with them. They reach places that might not get direct branches from the main sympathetic trunks.
What do these specific sympathetic fibers control? The ones in the skeletal nerves?
The source points to three main things: blood vessels, particularly in the skin and limbs, sweat glands all over the body, and the piloerector muscles.
Piloerector.
Yeah.
The ones that make your hair stand on end. Goosebumps.
That's them. So these fibers control blood flow to the periphery, temperature regulation via sweating, and yeah, goosebumps. The source even notes that something like 8% of all the fibers in an average skeletal nerve are actually these sympathetic fibers.
8%. That's quite a lot. Really highlights how widespread their influence is.
It does. Peripheral circulation, sweating. It's significant.
Okay, so that's the general wiring. How about getting signals to specific internal organs? Like T1 goes to the heart, T2 goes to the lungs, a neat map.
Not quite that neat, no. Because those preganglionic fibers can travel up and down the chain before synapsing, the mapping isn't strictly segmental like it is for, say, skin sensation. The source gives a general approximate guide, though.
Right.
Like fibers from T1 tend to go up to the head, T2 mostly to the neck, T3 through T6 generally supply the thorax, heart, lungs, esophagus. T7 through T11 tend to go to the abdomen, stomach, intestines, liver, kidneys. And T12 down to L2 mostly head down to the legs.
But approximate and overlapping.
Yes, definitely overlapping. It's not rigid boundaries.
Is there a reason for this general pattern?
The source links it back to how we develop as embryos. It's pretty cool, actually. Where an organ first started developing in the embryo often determines which part of the sympathetic chain sends nerves to it later on.
For example?
The heart. It actually starts developing up near the neck region in the embryo before it moves down into the chest. And guess what? It gets a lot of its sympathetic nerve supply from the cervical neck part of the sympathetic chain, which itself gets input from those upper thoracic spinal segments like T1 and T2.
Ah, so the nerves follow the organ's original location.
To some extent, yes. Same idea for the abdominal organs developing from regions corresponding to the mid to lower thoracic segments, hence their innervation from T7, T11. It's a neat link between development and adult anatomy.
Very cool. Now, the source flagged a really unique pathway in the sympathetic system, the one going to the adrenal medullae. It bypasses the usual synapse.
It does. This is a major exception. The preganglionic sympathetic fibers heading for the adrenal medullae, these two little glands sitting on top of your kidneys, they're different.
How so?
They travel all the way from the intermediolateral horn in the spinal cord, pass through the sympathetic chain, pass through the peripheral ganglia via splanchnic nerves, and go directly to the adrenal medulla without synapsing anywhere along the way.
So the first neuron, the preganglionic one, goes straight to the target cells in the adrenal gland.
That's right. It seems like it breaks the two-neuron rule, but the source explains why it doesn't, really. The cells inside the adrenal medulla that secrete hormones, they actually develop from the same embryonic nerve tissue as neurons themselves.
So the adrenal cells are like modified nerve cells.
Essentially, yes. They're considered modified postganglionic neurons. They've lost their axons, they're long fibers, but they're specialized to release signaling molecules, hormones, directly into the bloodstream instead of releasing a neurotransmitter across a tiny synapse onto just one cell.
And the hormones they release are epinephrine and norepinephrine.
Exactly. The source specifies it's about 80% epinephrine, also known as adrenaline, and 20% norepinephrine or noradrenaline released into the general circulation.
Which gives the sympathetic system a way to send signals everywhere via the blood.
Precisely. It's a hormonal reinforcement, way to broadcast the sympathetic message body-wide. We'll definitely talk more about its function later.
Okay, that covers the sympathetic side, the thoracolumbar outflow, the chains, the peripheral ganglia, the Gray-Rami detour, and that special adrenal pathway. Now, what about its counterpart, the parasympathetic nervous system? Where does this wiring start?
The parasympathetic system has a much different origin pattern. Its fibers leave the central nervous system from two distinct regions. The brainstem, traveling out with 4 specific cranial nerves, 3, 7, 9, X, and X, and the sacral region of the spinal cord, specifically segments S2 and S3, and sometimes S1 and S4.
Cranial nerves and sacral nerves, so not thoracic or lumbar like the sympathetic.
Right. That's why it's called the craniosacral outflow, much more spread out origins. Figure 61-3 in the source shows this layout.
And one of those cranial nerves, the vagus nerve, cranial nerve X, is particularly important here.
Hugely important. The vagus nerves are the real workhorses of the parasympathetic system. The source estimates they carry something like 75% of all parasympathetic nerve fibers in the entire body.
75%. Wow. Where do all those vagus fibers go?
Their reach is vast. They provide the parasympathetic supply to pretty much the entire chest and most of the abdomen. The source lists the major targets: heart, lungs, esophagus, stomach, the whole small intestine, the first half of the large intestine, proximal colon, liver, gallbladder, pancreas, kidneys, and the upper parts of the ureters.
So basically all the core functions in your torso are heavily influenced by the vagus nerve.
Very much so. Think digestion, heart rate regulation, largely vagal territory.
What about the other 3 cranial nerves carrying parasympathetic fibers? What do they do?
They handle parasympathetic functions in the head region. Cranial nerve the third, the oculomotor nerve, goes to the eye. It controls the muscle that constricts the pupil, pupillary sphincter, and the muscle that changes lens shape for focusing, ciliary muscle.
Okay.
Cranial nerve 7, the facial nerve, innervates glands. The lacrimal glands for tears, nasal glands for mucus, and the submandibular and sublingual glands for saliva.
Crying, sniffing, drooling,
Okay.
basically. And cranial nerve Nix, the glossopharyngeal nerve, primarily supplies the parotid gland, another major salivary gland. Got it. Head region glands and eye muscles. And then the sacral parasympathetic fibers. Where do they go?
These originate from those lower spinal cord segments, S2 and S3 mainly, and travel out in what are called the pelvic nerves. They supply the latter parts of the digestive tract, the descending colon and rectum, plus the urinary bladder, the lower parts of the ureters, and they play a key role in the reflexes causing erection of the external genitalia.
Okay, so covering the pelvic organs and lower gut, does the parasympathetic system also use that 2-neuron pathway, preganglionic and postganglionic, like the sympathetic?
Yes, it follows the same basic 2-neuron plan.
Right.
But, and this is a major anatomical difference, the location of the synapse is completely different.
Oh, so?
For the parasympathetic system, the source emphasizes that the preganglionic fibers are typically very long. They travel essentially uninterrupted almost all the way to the target organ itself.
Whoa, so the first neuron goes right up to the heart or the stomach wall.
Pretty much, yeah. With a few exceptions for some cranial nerves, that's the general rule.
So where's the second neuron, the postganglionic one?
This is the key distinction. The postganglionic neurons of the parasympathetic system are located right inside the wall of the organ they're controlling. The synapse between the long preganglionic fiber and the short postganglionic neuron happens within the target organ's tissue.
Inside the organ wall, so the postganglionic fibers must be tiny.
Extremely short, yes. The source says anywhere from a fraction of a millimeter up to maybe a few centimeters long. They just branch out locally from their cell body in the organ wall to innervate the nearby muscle or gland cells.
Okay, let's contrast that. Sympathetic. First synapse is usually far away from the target in the chain or a peripheral ganglion with long postganglionic fibers. Parasympathetic. First synapse is in the target organ's wall with super short postganglionic fibers.
You've nailed the key anatomical difference.
Does the source say why this structural difference might be important functionally?
It doesn't explicitly spell out the functional consequence right here in the anatomy section, but it's strongly implied by the structure. That sympathetic setup, synapsing further away, fibers traveling up and down the chain, diverging. It's perfectly built for coordinating broader, more widespread, sometimes body-wide responses. Think mass discharge.
Right.
The parasympathetic setup, with the synapse right in the target organ wall and those tiny local fibers, seems much more designed for very specific, targeted control of that individual organ's function without necessarily activating everything else nearby. It allows for fine-tuning.
Discrete control versus potential widespread mobilization. The anatomy dictates the strategy.
That's a great way to put it. The structure really enables the different functional roles of the two systems.
Okay, we've got the wiring maps. Now, how do these nerves actually talk to the organs? What are the chemical signals involved? The neurotransmitters.
Right. This comes down to the chemical messengers. The source focuses on two main ones used by the ANS to communicate with the effector cells. Acetylcholine, usually shortened to ACET, and norepinephrine, often called NE.
Acetylcholine and norepinephrine. And fibers are named based on which one they release.
Correct. Nerve fibers that release acetylcholine are called cholinergic. Fibers that release norepinephrine are called adrenergic.
And the source lays out a pretty clear pattern of which fibers are which, right? Figure 6104 helps visualize this.
Yes, there's a fundamental rule here. All preganglionic neurons, whether they belong to the sympathetic system or the parasympathetic system, are cholinergic. They release acetylcholine at that first synapse in the ganglion.
Okay, so ACET is the signal that excites the second neuron, the postganglionic one, in both systems.
Exactly. If you were to apply ACET or something similar to any autonomic ganglion, it would stimulate the postganglionic neurons of both the sympathetic and the parasympathetic nerves passing through it.
What about those postganglionic neurons, the ones that actually go to the organs? Do they use different transmitters?
This is where the main difference lies. According to the source, virtually all postganglionic neurons of the parasympathetic system are also cholinergic. They release acetylcholine onto their target organs, heart muscle, gut muscle, gland cells, etc.
Okay, so parasympathetic effects mostly driven by HA. What about sympathetic postganglionics?
Most postganglionic sympathetic neurons are adrenergic. They release norepinephrine onto their target organs.
So rule of thumb, parasympathetic output uses HA, sympathetic output uses NE. But wasn't there an exception we mentioned earlier when talking about the wiring?
Yes, absolutely. The crucial exception, the postganglionic sympathetic fibers that go to the sweat glands, and possibly a few specific blood vessels, are actually cholinergic. Even though they're part of the sympathetic system anatomically, they release acetylcholine, not norepinephrine.
The classic sweat gland exception. Sympathetic nerve, but uses HA. Okay, let me try to recap the chemistry.
Right.
All preganglionic neurons, both sympathetic and parasympathetic, release AC in the ganglia. Postganglionic parasympathetic neurons release AC onto the organs. Postganglionic sympathetic neurons mostly release NE onto the organs.
Right.
Except for the ones going to sweat glands and maybe some vessels, which release AC.
Perfect, you nailed it. So ACH is primarily the parasympathetic messenger at the target, and NE is primarily the sympathetic messenger at the target, with that key cholinergic sympathetic exception for sweating.
Okay. Now, how are these transmitters actually released at the nerve endings? Is it like a neat little synapse we see at skeletal muscles?
The source describes some similarities, but also important differences, especially for the sympathetic system. While some autonomic endings, maybe more so parasympathetic ones, might look a bit like smaller versions of the neuromuscular junction, many autonomic fibers don't form such distinct one-to-one connections.
How do they connect them?
Particularly for sympathetic fibers, they often just sort of touch or pass nearby the cells they influence. Sometimes the endings are just in the connective tissue near the target cells.
So not a classic synapse structure always. How do they release the transmitter reliably?
Instead of a single terminal button, these fibers often have multiple swellings or bulges along their length as they pass through the target tissue. These are called varicosities. Figure 61-4 shows this kind of beaded appearance.
Varicosities, like little storage depots along the nerve fiber.
Exactly. And these varicosities are where the neurotransmitter vesicles containing either AC or NE are synthesized and stored. They're also packed with mitochondria, the energy factories, to power this process.
And what triggers the release from these varicosities?
That part is pretty standard, similar to other synapses. When a nerve impulse and action potential travels down the fiber and reaches the varicosity, it depolarizes the membrane. This depolarization makes the membrane permeable to calcium ions.
Calcium rushes in.
Yep. Calcium ions, K++, flood into the varicosity from outside. And that influx of calcium is the critical signal that causes the vesicles filled with neurotransmitters to fuse with the varicosity membrane and dump their contents, ACH or Ne, into the space outside near the target cells. It's calcium-driven exocytosis.
Okay, calcium triggers release from the varicosities. Once the transmitter's out there, what happens to it? Let's follow acetylcholine first. How is it made and removed?
Right. Acetylcholine is synthesized right there in the nerve terminal or varicosity. The source mentions the ingredients are acetyl-CoA and choline, put together by an enzyme called choline acetyltransferase. Then it gets packaged into those vesicles.
And after it's released, does it hang around for long?
No, its action is designed to be very short-lived, just a few seconds usually. It gets broken down very rapidly in the synaptic space.
Broken down by what?
By an enzyme called acetylcholinesterase. This enzyme is hanging out right there in the local connective tissue near the nerve ending. It quickly splits the acetylcholine molecule into two pieces, an acetate ion and choline.
So the enzyme acts like scissors, cutting the AT8 and stopping its signal.
Exactly. Snip, snip, signal over. And it's efficient, too. The choline part that's released from this breakdown isn't wasted. It gets actively transported back into the nerve ending.
Recycled.
Yep, recycled to make new acetylcholine. The source notes this whole process: synthesis, release, rapid breakdown by acetylcholinesterase, and choline reuptake is basically the same mechanism used at the junction between nerves and skeletal muscles. It's a conserved strategy.
Okay, quick action, quick cleanup for HH. What about norepinephrine? How is it synthesized and how is it removed? How long does its signal last?
Norepinephrine synthesis is a bit more complex. It starts in the cytoplasm of the nerve ending, beginning with the amino acid tyrosine. Tyrosine gets converted to dopa, then dopa gets converted to dopamine.
Okay.
Then dopamine is transported into the storage vesicles. And the final step, converting dopamine into norepinephrine, actually happens inside the vesicle itself.
Inside the storage vesicle. Interesting. And wasn't there an extra step in the adrenal medulla?
Right. In those special cells in the adrenal medulla, there's one more enzymatic step inside the vesicle. About 80% of the norepinephrine gets converted further into epinephrine, adrenaline.
Got it. So Ne is released from a sympathetic nerve-ending varicosity. How is its signal stopped? Is there an enzyme like acetylcholinesterase waiting outside?
Not exactly the same. The source describes three main ways Ne is removed from the vicinity of the target cell, but one method is dominant for nerve endings.
What's the main one?
The most important way, accounting for maybe 50% to 80% of the removal, is active reuptake. The norepinephrine is pumped back into the adrenergic nerve ending it came from.
So the nerve ending sucks most of it back up.
Pretty much. It's an active transport process. This is the primary way the local signal is terminated quickly.
What happens to the rest?
Most of the remainder just diffuses away from the immediate area into the surrounding tissue fluid and eventually gets washed into the bloodstream.
And isn't there some enzymatic breakdown too?
Yes, a smaller amount gets destroyed by enzymes. The source mentions two key ones: monoamine oxidase, or MAO, which is actually located inside the adrenergic nerve endings themselves, so it can break down Ne that's reuptaken or hasn't been packaged, and catecholomethyltransferase, or COMT, which is found more broadly in various tissues and can break down Ne that diffuses away.
Okay, so reuptake is king for stopping the nerve signal quickly. How long does the effect of Ne typically last when released from a nerve?
Because of that efficient reuptake, the action of Ne released directly into a tissue by a nerve ending is usually quite brief, lasting only a few seconds.
But what about the Ne and epinephrine released from the adrenal medulla into the blood?
That's different. Once they're circulating in the bloodstream as hormones, they aren't subject to that rapid reuptake by nearby nerve endings. They get removed much more slowly, mainly by diffusion into tissues and eventual breakdown by enzymes like CRNT, particularly in the liver.
So they stick around longer.
Much longer. The source suggests their effects last maybe 10 to 30 seconds after secretion into the blood, with the activity gradually declining over 1 to several minutes. This more sustained action is really important for their role as hormones amplifying the sympathetic response.
Makes sense. Local nerve signal, quick on, quick off via reuptake. Hormonal signal.
Yeah.
Slower onset, longer duration, slower removal. Okay, so the transmitter ACH or Ne is released. It reaches the target cell membrane. What happens next? How does the cell actually get the message and respond?
This is where receptors come into play. Think of them as docking stations or locks on the surface of the target cell. The source describes them as specific protein molecules embedded in the cell membrane. The neurotransmitter acts like a key, binding to a specific spot on the outside of this receptor protein.
And that binding does something to the receptor protein.
Yes, it causes the protein to change its shape, a conformational change. That change in shape is the initial signal that tells the cell something has happened.
And how does the cell translate that shape change into an actual response, like muscle contraction or gland secretion? The source mentioned two main ways.
Right. The first way is by directly changing the cell membrane's permeability to certain ions. The receptor protein itself might be an ion channel, or it might be tightly linked to one.
So binding the transmitter opens or closes the gate.
Exactly. If the change opens channels for positive ions like sodium or calcium to flow into the cell, that usually makes the cell electrically excited, depolarized, which could trigger muscle contraction or nerve firing. If it opens channels for potassium ions to flow out of the cell, that makes the inside more negative, hypopolarized, which usually inhibits the cell.
Okay.
And sometimes just the change in ion concentration inside, like an increase in calcium, can directly trigger cellular processes, like activating contractile proteins in smooth muscle.
That's mechanism one. Direct ion channel changes. What's this second way receptor signal? Something about second messengers.
Right. Instead of directly opening a channel, the receptor, when activated by the transmitter binding on the outside, can interact with an enzyme that's attached to its inner part inside the cell membrane. Binding the transmitter activates or sometimes inactivates this intracellular enzyme.
And that enzyme then does something inside the cell.
Yes. The classic example given in the source involves norepinephrine binding to certain types of receptors. This binding can activate an enzyme called adenyl cyclase on the inside of the membrane.
And adenyl cyclase makes cyclic AMP.
Exactly. Adenyl cyclase takes ATP, the cell's energy currency, and converts it into cyclic adenosine monophosphate, or CAM-MP. The source explains that CAM-MP then acts as a second messenger. It's not the original signal, the transmitter, but it diffuses within the cell and triggers a whole cascade of other events activating other enzymes, changing gene expression that ultimately lead to the cell's specific response.
So the transmitter is the first messenger, CMP is the second. Are there others?
Yes, the source mentions other second messenger systems exist too, involving things like calcium ions interacting with a protein called calmodulin, or systems involving lipids in the membrane, like the phosphatidylinositol pathway. The key idea is the receptor activates an internal signaling cascade.
This really drives home a crucial point the source makes. It's not just the transmitter that determines the effect, excitation or inhibition. It's the type of receptor on the cell and what internal machinery that receptor is hooked up to.
Absolutely fundamental. The same neurotransmitter, say norepinephrine, can cause one organ to contract and another organ to relax, depending entirely on which specific type of adrenergic receptor that organ expresses and what signaling pathways are linked to it. You absolutely have to know the receptor type.
Okay, let's dive into those receptor types. The source starts with the receptors for acetylcholine, two main kinds.
Yes, muscarinic and nicotinic receptors. The names come from toxins that were historically used to differentiate them. Muscarine, from certain poisonous mushrooms, only activates muscarinic receptors. Nicotine, from tobacco, primarily activates nicotinic receptors. Acetylcholine itself, the natural transmitter, activates both types.
Where do we find the muscarinic receptors?
The source gives a clear rule. Muscarinic receptors are found on all the effector cells of the target muscle or gland cells that are stimulated by postganglionic cholinergic neurons.
So that includes all the target organs of the parasympathetic system.
Right, heart, gut, bladder, salivary glands, etc.
And the targets of those exceptional sympathetic fibers that release AGS, like the sweat glands.
Exactly. If the postganglionic fiber releases ASHI, the target cell will have muscarinic receptors. The source also notes these receptors typically work through G proteins and those second messenger systems we just talked about.
Okay. And the nicotinic receptors, where are they?
Nicotinic receptors are found in different locations. They're fundamentally different too. The source describes them as ligand-gated ion channels, meaning when ASHI binds, the receptor itself forms a channel that opens.
Where do we find them?
Primarily in two key places, according to this source. One, in the autonomic ganglia. They are located on the postganglionic neurons, both sympathetic and parasympathetic, where the preganglionic neuron synapses. So preganglionic HE excites the next neuron in line via nicotinic receptors. Two, at the neuromuscular junction and skeletal muscle. This is where somatic motor nerves release HE to make muscles contract. This shows a link in mechanism between the somatic and autonomic systems.
So let me try to map the HE receptors again. Preganglionic neuron releases AT binds to nicotinic receptors on the postganglionic neuron in the ganglion. Excites it. If it's a parasympathetic postganglionic neuron, it releases HE binds to muscarinic receptors on the target organ.
Perfect.
And if it's one of those special Sympathetic cholinergic postganglionic neurons like the sweat glands. It releases HE binds to muscarinic receptors on the sweat gland.
You've got the pattern exactly right. And understanding this distinction, nicotinic in ganglia, muscarinic on most target organs, is super important for pharmacology because drugs can be designed to target one type and not the other.
Right. Okay, now let's switch to the adrenergic receptors, the ones that bind norepinephrine, NE, and epinephrine, epi.
The source classifies these into two major families, alpha adrenergic receptors and beta adrenergic receptors.
And there are subtypes within those families.
Yes, there are at least alpha 1 and alpha 2 subtypes, and beta 1, beta 2, and beta 3 subtypes. These different subtypes are often linked to different intracellular signaling pathways, usually involving G proteins, and they can mediate different effects.
The source mentioned that NE and epi don't hit these receptors exactly the same way.
Correct. This is important, especially when thinking about the effects of hormones from the adrenal medulla versus neurotransmitter from nerves. Norepinephrine tends to excite alpha receptors more strongly than beta receptors. Epinephrine excites both alpha and beta receptors roughly equally.
So the effect on an organ depends on its mix of alpha and beta receptors.
Precisely. If an organ primarily has beta receptors, epinephrine will likely have a stronger effect on it than norepinephrine. If it's mostly alpha receptors, NE might be more potent or have similar potency. It depends on the specific receptor profile of the tissue.
The source provides Table 61:1 with examples of what alpha and beta receptors actually do. Can you give us a few key examples to make this concrete?
Sure, let's look at some classic effects. Alpha receptors, often alpha 1, these are heavily involved in causing smooth muscle contraction. Big examples include vasoconstriction, narrowing of blood vessels, particularly in the skin, gut, and kidneys. Also, dilation of the pupil in the eye by contracting the radial muscle, contraction of sphincters in the digestive tract, and making those piloerector muscles contract for goosebumps.
Okay, alpha mostly means constriction or contraction. What about beta?
Beta receptors are more varied. Beta 1 receptors. These are found in high concentration on the heart muscle. Stimulating them increases both the heart rate and the force of contraction. Think B1 for heart.
So beta 1 speeds up and strengthens the heart. Beta 2.
Beta 2 receptors often cause smooth muscle relaxation. Key examples include vasodilation, widening of blood vessels, and skeletal muscles getting more blood flow for exercise. Also, bronchodilation, relaxing the airways in the lungs, making it easier to breathe, and relaxation of the uterus.
Interesting. So sympathetic stimulation can cause blood vessels to constrict via alpha 1 in the gut, but dilate via beta 2 in the muscles.
Exactly. Same sympathetic system activating, same hormone like epinephrine potentially circulating, but opposite effects on blood vessels in different locations because they have different receptor subtypes. It powerfully illustrates that you can't just generalize.
What about beta 3?
Beta 3 receptors are mentioned as being involved in lipolysis, fat breakdown, and thermogenesis, heat production, particularly in adipose tissue.
This really drives home that receptor subtypes are critical. The source also touched on drugs that target these receptors selectively.
Yes, sympathomimetic drugs. These mimic sympathetic effects. Some are designed to hit specific subtypes. Phenylephrine is given as an example of an alpha agonist. It stimulates alpha receptors. Hence, its use as a decongestant constricts nasal blood vessels. Isoproterenol stimulates both beta 1 and beta 2, and albuterol is highlighted as a selective beta 2 agonist used in asthma inhalers to relax airways, beta 2 effect, without excessively stimulating the heart, which is mainly beta 1.
Selective targeting for therapy. Makes sense. Okay, we have the wiring, the chemicals, the receptors. Let's put it all together and look at the net effects of sympathetic versus parasympathetic stimulation on specific organs. Table 61:2 in the source seems key here.
It's a fantastic summary. It confirms what we've been saying. Both systems can excite or inhibit depending on the organ and receptors. Sometimes they work in opposition, reciprocal control, but sometimes one system just dominates a function. The source warns there's no simple rule like sympathetic always excites. You really have to learn the effects organ by organ.
Okay, let's walk through some major examples from the source. The eyes.
Right. Two main functions controlled by the ANS here: pupil size and lens focus. Pupil. Sympathetic stimulation contracts the radial muscle of the iris, causing the pupil to dilate, mydriasis. Parasympathetic stimulation contracts the circular sphincter muscle, causing the pupil to constrict, meiosis. This is a classic push-pull reciprocal action.
Parasympathetic for bright light, Sympathetic for dim light or excitement.
Exactly. And lens focusing, lens focus accommodation. This is almost entirely under parasympathetic control. When the parasympathetic system stimulates the ciliary muscle in the eye, it contracts. This relaxes the ligaments holding the lens, allowing the lens to bulge and become more convex for focusing on near objects. Sympathetic has little effect here.
Got it. Eyes. Pupil is dual control. Focus is parasympathetic. What about glands, like salivary glands, tear glands, gut glands?
For glands involved in digestion and lubrication in the head and upper GI tract, like nasal, lacrimal, tear, salivary, and stomach glands, the parasympathetic system is generally the strong stimulator. It causes them to produce copious, thin, watery secretions. Think mouth-watering.
And sympathetic stimulation.
Sympathetic stimulation tends to cause a smaller volume of secretion, and it's often thicker, more concentrated with enzymes or mucus. But there's a twist. Sympathetic nerves also cause vasoconstriction, narrowing the blood vessels supplying the glands. So even if sympathetic signals directly stimulate the gland cells a bit, the reduced blood flow can actually decrease the overall rate of secretion. It's complex.
Right. And then there's the sweat glands, the big exception again.
The sweat glands responsible for cooling the body? Yes. These are stimulated to produce copious sweat by the sympathetic system, but remember, those sympathetic fibers are cholinergic, release ACC onto muscarinic receptors. Parasympathetic system has no effect on these sweat glands.
So sweating is anatomically sympathetic, but chemically cholinergic. What about the other type of sweat glands, the apocrine ones?
The apocrine glands, like in the armpits, associated with odor, those are also stimulated by the sympathetic system, but via the usual mechanism. Adrenergic fibers releasing norepinephrine. Parasympathetic has no effect here either. So even different types of glands under sympathetic control use different chemical signals.
Wow. Okay. How about movement in the digestive tract? Motility.
The ANS influences gut movement largely by modulating the gut's own intrinsic nervous system, the enteric plexus. Generally, parasympathetic stimulation increases overall GI activity. It boosts peristalsis, the waves pushing food along, and relaxes sphincters, helping food move through faster. It also increases secretion.
And sympathetic, the opposite.
Usually, yes. Sympathetic stimulation generally inhibits peristalsis and increases the tone tightness of sphincters. This slows down the movement of gut contents, potentially leading to constipation. It can also decrease secretion somewhat. The source does note that normal gut function isn't super dependent on sympathetic input, unlike the crucial role of parasympathetic tone.
Okay. The heart is often used as the prime example of opposing control, right?
It really is. Sympathetic stimulation acts like pressing the accelerator. It increases both the heart rate and the force of contraction, especially via beta-1 receptors, making the heart pump more blood faster, essential for exercise or stress.
And parasympathetic.
Parasympathetic stimulation, mainly via the vagus nerve, is the brake pedal. It decreases the heart rate and also reduces the force of contraction, particularly in the atria. This allows the heart to rest and conserve energy, and very strong vagal stimulation can actually stop the heartbeat for a short time.
Dramatic. What about blood vessels throughout the body? Systemic blood vessels.
Here, the sympathetic system is overwhelmingly dominant for control. Most blood vessels, especially those supplying the abdominal organs and the skin, are constricted by sympathetic stimulation, mostly via alpha receptors.
And parasympathetic effect on blood vessels.
Generally, very little or none on most systemic vessels. While parasympathetic nerves might dilate vessels in a few specific areas, like salivary glands or external genitalia, they don't have widespread control over vascular resistance like the sympathetic system does.
So blood pressure regulation is mainly a sympathetic game.
In terms of actively changing vessel diameter body-wide, yes. Arterial pressure depends on how much blood the heart pumps out, cardiac output, and how much resistance the vessels offer, peripheral resistance.
So sympathetic increases both.
Correct. Sympathetic stimulation increases both heart pumping and peripheral resistance via vasoconstriction, leading to a significant acute rise in blood pressure. The source clarifies it has less direct impact on long-term pressure unless it affects kidney function.
And parasympathetic effect on pressure.
Moderate parasympathetic activity primarily just slows the heart, reducing cardiac output. Since it doesn't affect most vessels, the result is usually only a slight decrease in pressure. But again, very strong vagal stimulation that stops the heart can cause a temporary severe drop in pressure.
Got it. The source also quickly lists some other effects.
Yeah, just brief mentions, like sympathetic, generally inhibiting ducts and tubes, like bile ducts, ureters, bronchi, while parasympathetic tends to excite them. And it flags the important metabolic effects of sympathetic stimulation, things like releasing glucose from the liver, breaking down glycogen, increasing muscle strength, boosting the overall metabolic rate, BMR, and even increasing mental alertness. And finally, the roles in sexual function. Erection, mainly parasympathetic, ejaculation involving sympathetic components, just highlighting the broad reach.
Okay, so that covers the direct nerve effects. But we also talked about the adrenal medulla acting like a hormonal amplifier for the sympathetic system. Let's revisit that.
Right. This is a critical part of the sympathetic response. When those special preganglionic sympathetic nerves stimulate the adrenal medulla, it dumps large amounts of epinephrine, about 80%, and norepinephrine, about 20%, directly into the bloodstream.
And these hormones circulate everywhere. What do?
They travel via the blood to basically all tissues, and they bind to the same alpha and beta adrenergic receptors that the nerve-released norepinephrine binds to. So they produce almost the same effects as if you stimulated sympathetic nerves everywhere at once.
Almost the same. What's the key difference then?
The duration of action. Nerve-released NE gets sucked back up very quickly, so its effect is short. But these hormones circulating in the blood, they get removed much more slowly, mainly broken down in the liver and other tissues.
So their effects last longer.
Much longer. The source says 5 to 10 times longer than direct nerve stimulation. The effects might peak within 10 to 30 seconds, but can persist, declining over maybe 2 to 4 minutes. This provides a more sustained, widespread sympathetic boost or background hum.
Does the source compare the specific effects of the circulating norepinephrine versus the circulating epinephrine, since they hit receptors differently?
Yes, it does. Circulating norepinephrine primarily acts like widespread sympathetic nerve stimulation. Strong vasoconstriction, alpha effect, increased heart activity, beta-1 effect, etc. This really cranks up the total peripheral resistance and thus elevates arterial pressure significantly.
And epinephrine.
Epinephrine also stimulates the heart strongly, beta-1, and causes some vasoconstriction, alpha. But because it hits beta receptors harder overall, especially beta-2, it causes more pronounced vasodilation in places like skeletal muscle. So the source notes that while both raise blood pressure, epinephrine might raise it less than norepinephrine does, but it increases cardiac output, the amount of blood pumped, more due to the stronger heart stimulation combined with reduced resistance in muscle beds.
And epinephrine has stronger metabolic effects too.
Much stronger, yes. The source states epinephrine has maybe 5 to 10 times the metabolic impact of norepinephrine. It can significantly increase the whole body's basal metabolic rate, sometimes up to 100%. It's also much more potent at stimulating glycogen breakdown in the liver and muscle, releasing glucose into the blood for energy. It really revs up overall cellular activity and excitability.
So the adrenal medulla really serves to back up and enhance the direct nerve signals.
Absolutely. It's a support system. Usually the adrenals get stimulated whenever there's a general sympathetic discharge. This gives you a dual mechanism: direct, fast nerve stimulation and slower, longer-lasting, widespread hormonal stimulation.
Provides a safety factor too.
Exactly. If nerve pathways are cut, the hormones can still provide some sympathetic function. If the adrenals are removed, the nerves can usually compensate. Plus, the hormones can reach cells that might not even have direct sympathetic nerve connections, like epinephrine boosting the metabolism of almost every cell in the body. It's a really elegant backup and amplification system.
Powerful synergy. Okay, let's shift perspective a bit and look at the operational characteristics. How fast do these systems need to fire, and what's this idea of tone? The source compares the firing rate needed to skeletal muscle.
Yeah, it's quite different. Autonomic targets like smooth muscle or glands don't need the super high-frequency firing rates that skeletal muscle does to get a full response. For autonomic effects, the source says even just one nerve impulse every few seconds might be enough to maintain a baseline level of activity. Full activation might only require 10 to 20 impulses per second.
Whereas skeletal muscle needs hundreds.
Sometimes 50 to 500 or more impulses per second for maximal smooth contraction. It reflects the generally slower nature of smooth muscle and gland responses compared to fast-twitch skeletal muscle.
And this relates to the concept of tone. What exactly is sympathetic or parasympathetic tone?
Tone just refers to the fact that under normal resting conditions, both the sympathetic and parasympathetic systems are usually firing continuously at a low baseline rate. They're almost always on to some degree.
Why is that constant background activity important?
It's incredibly useful because it allows a single system to control an organ's activity in both directions, up or down from that baseline.
How does that work?
The source uses the example of sympathetic tone on blood vessels. Normally, sympathetic nerves fire enough to keep arterioles constricted to about half their maximum diameter. That's the baseline tone. From there, if the sympathetic system fires more, the vessels constrict further. If it fires less than the baseline rate, the vessels dilate. So just by varying its firing rate relative to the resting tone, the sympathetic system can cause either vasoconstriction or vasodilation.
Without needing a separate vasodilator system.
Exactly. Tone provides that bidirectional control capability within one system.
Is there an example for parasympathetic tone?
Yes, the source mentions parasympathetic tone in the gut. The vagus nerve normally provides a baseline level of stimulation that's necessary for normal digestive motility. If you cut the vagus nerves, the gut loses this tone, becomes sluggish, atonic, and you get constipation. The brain can then modulate this tone, increase vagal firing to promote digestion, decrease it to slow things down.
Does the adrenal medulla contribute to this baseline tone?
Yes, the source explicitly points out that the normal low-level resting secretion of epinephrine and norepinephrine from the adrenal medulla contributes significantly to the overall background sympathetic tone, helping maintain things like normal blood pressure.
What happens if this tonic input is suddenly lost? Like if you cut the nerve supplying an organ?
The immediate effect is that the organ loses that baseline drive. So if you cut the sympathetic nerves to blood vessels, they immediately dilate because the tonic constricting signal is gone, loss of tone.
But the source describes a fascinating adjustment that happens over time.
Yes, this is really crucial. Over days or weeks, the tissue itself starts to compensate. The smooth muscle, for example, might develop increased intrinsic tone, becoming more contractile on its own due to internal chemical changes, even without nerve input. More dramatically, the tissue becomes much, much more sensitive to any remaining signal.
This is the denervation supersensitivity.
Exactly. The organ becomes hypersensitive to the neurotransmitter it's been deprived of. The source uses figure 61-5 to illustrate this with blood flow in an arm after the sympathetic nerves are cut.
What does the figure show?
Okay, before cutting the nerves, baseline blood flow is normal, and injecting a small test dose of norepinephrine causes a small dip in flow. Immediately after cutting the nerves, flow shoots up because the constrictor tone is gone. Then, over weeks, flow comes back down somewhat as the vessel develops some intrinsic tone. But now, if you inject that same small test dose of norepinephrine,
It has a much bigger effect.
a huge effect. The blood flow plummets way more than it did originally. The source says the vessel might be 2 to 4 times, sometimes even more than 10 times, more responsive to norepinephrine than it was before denervation. Super sensitive.
Wow. Why does that happen? How does the tissue become so much more sensitive?
The main mechanism proposed in the source is upregulation of receptors. Basically, when the target cell isn't getting its usual neurotransmitter signal anymore, it responds by putting many more receptor proteins onto its surface membrane.
More docking stations.
Exactly. So now, even a tiny amount of transmitter floating around in the blood or a small injected dose can bind to way more receptors and trigger a much larger response. It's a powerful adaptive change by the target tissue itself.
Incredible. The cell literally changes its sensitivity based on the input it's not getting. Okay, so the ANS has this baseline tone, it allows fine-tuning, and the target tissues adapt if tone is lost. A lot of this regulation happens automatically for reflexes, right?
Yes, autonomic reflexes are the workhorses of moment-to-moment regulation. Subconscious inputs trigger pre-programmed outputs.
The source gave several examples. Cardiovascular ones.
The baroreceptor reflex is the classic. Stretch receptors in your major artery sense if blood pressure gets too high. They signal the brainstem. The brainstem then reflexively turns down sympathetic output and turns up parasympathetic output to the heart and vessels.
Result.
Heart slows down, vessels relax a bit, pressure comes back down. It's a negative feedback loop keeping pressure stable.
What about gastrointestinal reflexes?
The source mentions reflexes at both ends, upper end. Just smelling or tasting food triggers signals to the brainstem, which then uses parasympathetic nerves, like the vagus, to tell your salivary glands and stomach to start secreting digestive juices, getting ready. Lower end. When the rectum fills and stretches, sensory signals go to the sacral spinal cord, triggering a parasympathetic reflex back to the colon and rectum, causing contractions and relaxation of sphincters for defecation.
Built-in programs. Any other examples?
Yep. The urinary bladder emptying mixturition reflex is similar. Bladder stretch triggers a sacral cord reflex, causing bladder contraction and sphincter relaxation. Sexual reflexes are mentioned too, initiated by psychic stimuli or local sensation processed in the sacral cord, leading to erection, mostly parasympathetic, and then ejaculation involving sympathetic components. The source also briefly lists reflexes controlling pancreatic secretion, gallbladder emptying, kidney excretion, sweating, blood glucose, showing just how many bodily functions rely on these automated loops.
These reflexes are like the body's standard operating procedures for essential tasks. Now, thinking about how the systems get activated, does the sympathetic system always fire as a single unit, or can it be more selective?
The source makes a distinction here. The sympathetic system is capable of, and famous for, firing in what's called a mass discharge.
When would that happen?
Typically when the hypothalamus gets strongly activated, usually by intense fear, sudden severe pain, or extreme excitement, the classic fight or flight trigger.
And mass discharge means?
It means pretty much the entire sympathetic system fires off together all at once as a coordinated body-wide response. This generates the full-blown alarm or stress reaction.
What are the effects listed for this mass discharge?
It's a whole suite of changes designed for intense activity. Arterial pressure shoots up, blood flow gets shunted away from non-essential areas like the gut and skin and redirected to active muscles in the brain. Cellular metabolism and BMR increase across the body. Blood glucose levels rise, providing fuel. Muscle strength increases. Mental alertness heightens. Even blood clotting speeds up, potentially preparing for injury.
Basically mobilizing every resource for maximum physical exertion or dealing with a major threat.
Exactly. It's the physiological basis of the fight-or-flight reaction. Strong emotions like rage, mediated via the hypothalamus, can trigger this massive sympathetic outflow.
But it's not always all or nothing like that. Can the sympathetic system act more locally?
Yes, it absolutely can activate in more isolated portions. The source gives key examples. 1. Heat regulation. Controlling sweating and skin blood flow involves sympathetic nerves, but these can be adjusted for temperature control without necessarily triggering a full-body stress response. 2. Local reflexes. Sometimes sensory input can trigger a localized sympathetic response, like heating one patch of skin might cause vasodilation and sweating just in that area via reflexes involving only local sympathetic ganglia or spinal cord segments. 3. GI reflexes. Some gut reflexes might involve sympathetic pathways that just loop between the gut, the nearby paravertebral ganglia, and back to the gut without even going up to the spinal cord.
So it has both modes, full body alarm and more localized adjustments. How does the parasympathetic system typically activate? Mass discharge too?
Generally, no. The parasympathetic system usually activates in more specific, localized ways. A parasympathetic reflex is more likely to affect, say, just heart rate or just salivary secretion or just bladder emptying without involving the whole system.
But didn't the source say related functions can happen together?
Yes, while typically localized, functions that are physiologically related often are activated in concert. For example, during digestion, salivary, gastric, and pancreatic secretions might all increase together due to coordinated parasympathetic activity. And the reflexes for rectal emptying and bladder emptying are closely linked and can often trigger each other. So, not a body-wide mass discharge, but functionally related clusters can be activated together.
That makes sense. Discrete control, but coordinated where needed. Okay, that gives us a good feel for the operational modes. Finally, let's look upstairs the control centers in the brainstem and hypothalamus and how higher brain areas fit in.
Right. The source points to specific clusters of neurons within the brainstem in the reticular substance and areas like the tractus solitarius nuclei, located in the medulla, pons, and midbrain as being key control centers for specific autonomic functions. Things like regulating arterial pressure, heart rate, GI activity, bladder contraction. These fundamental controls are wired in the brainstem.
The source mentioned experiments cutting the brainstem to locate these centers.
Yeah, it's a classic way to figure out what different levels do. If you cut the brainstem above the middle of the pons, basic blood pressure control can still operate, suggesting centers below that cut are sufficient. But if you cut below the medulla, pressure plummets, indicating essential pressure control centers are in the medulla.
And these brainstem centers aren't acting alone. They get input from higher-ups.
Critically important, yes. The hypothalamus, sitting above the brainstem, exerts major control over many of these brainstem autonomic centers. For example, stimulating certain parts of the hypothalamus, especially the posterior regions, can trigger massive sympathetic output and huge increases in blood pressure. Other hypothalamic areas regulate body temperature, appetite, water balance, salivation, bladder function, often by sending signals down to modulate the brainstem centers.
So the hypothalamus is like a master regulator coordinating autonomic output with other drives and needs.
That's a good way to think of it. And it doesn't stop there. Even higher centers, like the cerebral cortex, especially the limbic cortex, our emotional brain feeds signals down, primarily through the hypothalamus.
So thoughts and emotions can influence the hypothalamus, which influences the brainstem, which controls the autonomic output.
Exactly. It forms this integrated network where the hypothalamus, brainstem reticular areas, and the ANS work together to mediate not just basic physiology, but also our behavioral responses to the world. Our mental and emotional state has a direct line down to our guts, heart, and glands via this pathway.
And this connection leads to the idea that chronic stress or strong emotions, mediated through the brain's influence on the ANS, could actually contribute to physical diseases.
The source explicitly mentions this possibility. It suggests that prolonged or excessive influence from higher brain areas altering ANS function might play a role in conditions like peptic ulcers, related to the stomach acid secretion and blood flow, constipation, gut motility, heart palpitations, maybe even contributing to the risk of heart attacks. It really highlights that mind-body connection, how psychological factors can have real physical consequences mediated through the autonomic nervous system.
That brings us neatly to the final major section outlined in the source pharmacology. How can we use drugs to interact with this incredibly complex system?
This is obviously a huge area in medicine because the ANS controls so many vital functions. Drugs can be designed to target almost any step in the process, mimicking transmitters, blocking transmitters, hitting specific receptor subtypes, interfering with synthesis or breakdown.
Let's start with drugs that mimic sympathetic action, the sympathomimetics.
Right. These drugs stimulate the adrenergic effector organs, the targets of sympathetic nerves. Injecting norepinephrine or epinephrine themselves works, but as we said, their effect is very short. Many other drugs are developed for longer action, maybe 30 minutes to a few hours.
Including those selective ones we mentioned.
Exactly. The source reiterates examples like phenylephrine, mostly alpha agonist, isoproterenol hits beta-1 and beta-2, and albuterol, selective for beta-2. These allow for more targeted effects.
Are there drugs that cause sympathetic-like effects indirectly?
Yes. Some drugs work not by stimulating receptors themselves, but by causing the sympathetic nerve endings to release more of their stored norepinephrine. The released NE then acts on the receptors. Ephedrine, tyramine, and amphetamine are given as examples of these indirectly acting sympathomimetics.
Okay. What about drugs that block sympathetic activity?
These can interfere at various points along the pathway. The source lists several mechanisms. 1. Block any synthesis or storage inside the nerve ending, example reserpine. 2. Block the release of NE from the nerve ending, example daquanethidine. 2. Block the release of NE from the nerve ending, example. 3. Block the adrenergic receptors themselves. This is a huge category. Alpha blockers can be non-selective, blocking both alpha-1 and alpha-2, like phenoxybenzamine or phentolamine, or selective, like prazosin for alpha-1 or yohimbine for alpha-2. Beta blockers. Also non-selective, blocking beta-1 and beta-2, like propranolol, or selective, like atenolol, metoprolol, focusing on beta-1, primarily used for heart conditions. Beta blockers. 4. Block transmission through the autonomic ganglia. Since sympathetic signals pass through ganglia, blocking ganglia also blocks sympathetic output. Drugs like hexamethonium, though these block parasympathetic too.
A whole arsenal for dampening sympathetic effects. Now, what about drugs acting on the cholinergic side, mimicking parasympathetic actions?
These are parasympathomimetics, or cholinergic drugs. Again, injected HEA is broken down too fast to be useful. But other drugs can directly stimulate the muscarinic receptors on target organs. Pilocarpine and methacholine are examples given.
Are there drugs that boost the effect of the body's own acetylcholine?
Yes, those are the anticholinesterase drugs. They work by inhibiting the enzyme acetylcholinesterase, the cleanup crew that normally destroys HRS. By blocking the enzyme, they allow naturally released HE to hang around longer and have a stronger, more prolonged effect at cholinergic synapses.
Like turning up the volume on the parasympathetic signal.
Exactly. The source lists neostigmine, pyridostigmine, and aminonium as examples, noting these also work at the skeletal neuromuscular junction.
And drugs that block cholinergic effects at the target organs.
These are the antimuscarinic drugs. They block the action of HES specifically at the muscarinic receptors found on parasympathetic target organs and those cholinergic sympathetic targets like sweat glands. Atropine, homatropine, and scopolamine are the key examples.
Importantly, these don't block the other type of AC receptor.
Correct. The source emphasizes that these antimuscarinic drugs do not affect the nicotinic acetylcholine receptors located in the autonomic ganglia or at the skeletal muscle junction. Very specific blockade.
Okay, one last category. Drugs that act directly at the autonomic ganglia, affecting the postganglionic neurons themselves, stimulating them first.
Well, the natural stimulus is AC acting on nicotinic receptors, as we said. So injected AC can stimulate postganglionic neurons of both systems. The classic drug example here is nicotine itself.
Nicotine stimulates the ganglia.
Yes, it activates those nicotinic AC receptors on the postganglionic neurons. That's why they're called nicotinic receptors. The effect of nicotine can be complex because it simultaneously excites both sympathetic and parasympathetic postganglionic neurons. You might get increased heart rate, sympathetic, but also increased gut activity, parasympathetic, at the same time.
Messy. What about drugs that block transmission at the ganglia?
These are the ganglionic blocking drugs. They prevent the signal from passing from the preganglionic neuron to the postganglionic neuron, effectively shutting down outflow in both sympathetic and parasympathetic systems at that level. Tetraethylammonium, hexamethonium, and pentylenium are examples.
So they block everything.
Pretty much all autonomic outflow. The source notes they were sometimes used clinically to achieve strong sympathetic blockade, like lowering blood pressure drastically, because the effects of blocking the sympathetic system often overshadow the effects of blocking the parasympathetic. But they cause widespread side effects and are hard to manage, so their use is limited.
That's an incredibly detailed map of the pharmacology. It seems like you can target almost every step: synthesis, storage, release, receptors, breakdown.
It really does. It underscores how complex the system is, but also how understanding these details allows for interventions, whether therapeutic or, unfortunately, toxicological, that can have very specific or very broad effects on our physiology.
Wow. Okay. We have really gone deep into this source material on the autonomic nervous system today. It feels like we've covered a huge amount of ground.
We certainly have.
We started with just defining it, that incredible speed. That we mapped out the wiring, the sympathetic thoracolumbar outflow, the parasympathetic craniosacral outflow, those key anatomical differences.
Right, the ganglia locations, the fiber lengths.
Then the chemical signals AC and NE, and the crucial exceptions, and the receptors, muscarinic, nicotinic alpha subtypes, beta subtypes, how they determine the actual effect on different organs.
We looked at those specific organ effects, the push and pull or dominance of the two systems, the amplifying role of the adrenal medulla's hormones, adding that sustained, widespread component.
And the dynamics, the concept of baseline tone, how it allows for up-down regulation, and that fascinating denervation supersensitivity, the tissue adapting to lost input.
Don't forget the reflexes, the built-in automation, and the control centers, brainstem, hypothalamus, and the influence from the emotional brain.
And finally, that whole landscape of pharmacology, how drugs can manipulate the system at so many different points.
It's been a comprehensive tour, really digging into the mechanics and concepts laid out in this source, aiming for that deep foundational understanding.
So, for you listening, why does wading through all this detail matter? What's the payoff?
Well, getting the ANS at this level gives you, I think, a really profound insight into how your own body works automatically. It explains so much about health and disease, why stress hits your gut, why your heart races when you're scared, how your body copes with emergencies, how blood pressure stays stable or doesn't.
And understanding medications, too.
Absolutely essential. So many common drugs, from asthma treatments to drugs for blood pressure, anxiety, digestion, they work by targeting parts of the ANS. Understanding the system is key to understanding how those drugs work, why they have side effects. It's really a shortcut to deeper physiological insight.
It definitely feels like peering under the hood and seeing this incredibly complex, constantly running machinery that we normally just take for granted.
That's a great analogy. It's humming away all the time.
So let's end with a final thought. Building on that idea of tone and supersensitivity, we talked about how removing nerve input changes the baseline tone and makes tissues hypersensitive. So consider this. What if the influence isn't complete removal, but something more subtle and chronic, like persistent psychological stress, maybe chronic low-grade inflammation, maybe consistently poor sleep? Could these kinds of things subtly shift the baseline tone of the sympathetic or parasympathetic system over long periods? Or maybe gradually change the responsiveness of the target organs, altering receptor numbers, perhaps even without any obvious nerve damage?
That's a really interesting and complex question. It pushes beyond simple reflexes into long-term adaptation. Could living with a slightly chronically elevated sympathetic tone, for example, gradually change how your heart or blood vessels or immune system respond to everyday challenges down the line? Does the system recalibrate its normal?
Something to think about, definitely. How these constant background signals and the tissues listening to them might interact and change over a lifetime. Food for thought as you encounter ideas about stress, chronic illness, and how our bodies regulate themselves in other contexts. Thanks for diving deep with us today.